Alrighty. So today we're going to dive into some essential concepts about blood involving blood composition. Then we'll walk through how red blood cells are removed and how heme is broken down. Finally, we'll cover three mechanisms of hemostasis, and then we'll look into the coagulation cascade. So I'll be sketching just to help us out understand a little bit. So this first part is going to be about blood composition. So here I have a test tube showing us the composition of blood. So the full test tube is 100% of your blood, and so the composition of your blood is made up of two main parts. First one is plasma, and then the other one is like formed elements. And so we'll talk about that in a minute. So plasma makes up about 55% of your blood volume, and forced elements, which are the cells and cell fragments, these make up about 45% of your blood volume. And that 1% is called the buffy coat. So the formed elements include red blood cells, which carry oxygen, and white blood cells, which defend against infections, and platelets, which help with blood clotting. So that is your blood composition. So this next part is going to be about red blood cell removal and heme breakdown. So red blood cells typically live about 120 days. After that, they're removed by macrophages, primarily in the spleen, liver, and the bone marrow. So inside the red blood cell is hemoglobin, which gets broken down into two distinct parts. And so here I have, can't really see that very well. So here I have a red blood cell, and the red blood cell gets broken down into heme and globin. Okay, so it gets broken into those two major parts. The globin portion is then broken into amino acids. And those amino acids are then recycled, reused in the body. The heme portion gets broken down into first iron, which is then stored and recycled or transported, I guess, stored or transported for reuse in the body, so it's recycled as well. And then heme gets broken down into uh, porphyrin ring, which then gets converted into an unconjugated bilirubin, and then it travels to the spleen where it gets conjugated and excreted into bile. From there, it travels through the intestines and it eventually gets eliminated in feces. And this is actually what kind of causes that brown coloring in feces. Righty. Now we're going to talk about hemostasis, which is the body's way of stopping bleeding. And there are three main steps. First two are vasoconstriction and platelet plug formation. Those are pretty easy. And then the last one is coagulation. So vasoconstriction happens when the vessel gets injured and smooth muscle outside the vessel or in the vessel walls constricts or forces it to narrow, um, causing reduced blood flow, right? And so we see here, these are red blood cells, and then the green are platelets. Okay, and the arrow indicates vasoconstriction. The next step is platelet plug formation. This one is quite important because it has to do with the first steps of clotting, right? Preventing the blood from coming. So we have exposed collagen fibers at the injury site. These platelets adhere to it thanks to a protein called von Willebrand factor. Okay, these platelets then activate and release chemicals like ADP to attract more platelets. And then this leads to platelet aggregation, forming a temporary clot or a plug to slow the bleeding. Not stopping it, but slowing it. Then this next step is coagulation cascade. Okay, this one, there's three distinct pathways in coagulation cascade. The entire step stabilizes the platelet plug with a mesh of fibrin. Okay, so it replaces it something temporary with something a little more permanent. And it does this with three specific pathways. The intrinsic pathway, which is triggered by the damage of the blood vessel itself, and that is going to include factors 12, 11, and 9, as well as 8. Okay, and the extrinsic pathway is triggered by tissue factor, released from outside of the vessel, and it involves factor 7, as we can see up here. Factor X or factor 10 activate, or both pathways lead to the common pathway, starting with factor 10. Okay, factor 10 activates thrombin and prothrombin. Then thrombin converts fibrinogen to fibrin, which forms a strong mesh over the platelet plug, stabilizing the clot. Right, so this last section is fibrinolysis, or dissolving of the clot. And this occurs after the vessel is healed and the clot is no longer needed. And so that's where fibrinolysis comes in. So I have a quick little diagram. And once everything's all healed up, we have the clot preventing any blood flow, right? And so plasminogen, which is found within the fibrin mesh, is activated by TPA, or tissue plasminogen activator, which then forms plasmin, an enzyme that digests fibrin and dissolves the clot. Okay, and so it does that by releasing TPA, which then converts the plasminogen to plasmin, reducing the amount of fibrin mesh because it's being digested by plasmin here, causing more blood flow once the clot has been healed or the injury has been healed and the clot needs to be done. So this cleanup process prevents unnecessary clotting and allows the vessel to return to normal, also known as hemostasis, right? And so basically, to summarize everything today, blood contains plasma and formed elements like red blood cells. Okay, red blood cells are removed by macrophages, and their heme is broken down into iron and bilirubin. Okay, and then that gets converted into bile and then into feces. Hemostasis has three important steps: vasoconstriction, platelet plug formation, and coagulation. The coagulation cascade includes intrinsic, extrinsic, and common pathways, including factors 12, 11, 9, and 8. For the intrinsic and extrinsic is 7. And then the common pathway includes 10, factor 10. And then finally, TPA activates plasminogen into plasmin to remove the clot when it is no longer needed. So, thanks for watching.